Hey.
Hey, nice haircut.
Thank you.
Hello there.
Hi, how are you?
How are you? Yeah, I'm good.
Good, thank you.
How is the UK now?
It's okay. We've just come out of another lockdown, so yeah, but vaccines are coming, so hopefully maybe sometime next year it'll be okay. How's everything at your end?
In Singapore it's still okay, but I'm moving to the US next week.
I was going to say, yeah, congratulations.
Yeah, which is not a good time at all. Yes.
Moving to Boston, right?
Yeah, I'm moving to Boston.
That's a nice city.
You ever been there?
Yeah, I used to live in Philadelphia, so
Oh, okay.
I went to Boston a few times. It's nice.
Yep.
Very European feel. You're gonna have to tell me how to pronounce your name.
Yes.
Otherwise, me and Jen between us have screwed up everyone's names in this so far.
So you can introduce me as Anh Phương Lê.
Anh Phương Lê.
Yeah. Just call me Phương for generally. Yep.
Yeah, got it. Okay, I'll try my best.
Yeah. And then don't worry about the introduction too much.
Okay.
Yes.
No worries.
Yeah.
Do you want to test out your presentation?
Yeah, sure.
Yeah.
How do I share this?
You should be able to share all of it. Okay.
I don't know. I couldn't. Oh yes, this is the share screen. I think I will share my main screen because if I share.
Yeah, whatever works for you.
And then I don't want it to block this. Yes. So if I do it like this, it would be.
Yeah, looks good.
Yep. So the movie is okay.
Yeah, it's playing well. Cool.
There's one more movie. Then this. I hope this is okay. It's okay.
Yeah, that's good.
Okay,
yeah,
nice.
Great. Thanks a lot.
Yep. Okay.
Do you have a preference whether you want to go first or second, or you don't mind?
I don't.
Okay. Becky, is the YouTube going okay?
Let me check. Hang on. It's just I'm gonna mute this. Yeah, it's all good.
Okay. And the sound's okay as well, yeah?
Yeah, it was great.
Okay. Do you know whether you need to, when we finish up, do you need to end it before I close the meeting? Or can I just close the meeting and it will end the stream?
I think it will end it. I think
when I don't know, because I don't know, are you able to end it then?
Yeah, you can do it from the YouTube account.
Okay. I don't know whether because Jen started it, she also needs to end it.
No, I think we're all right.
Okay. Let me tweet it out. No.
you excited for tomorrow?
What's happening tomorrow? Oh yeah. I forgot. Yeah. It's just another day at this point.
Oh. Are we going out for a lunch or something? A socially distanced lunch takeaway?
Yeah, we'll have to, yeah.
Come in.
We'll talk after this. We'll decide afterwards. Yeah, I should have realized when we were saying the dates about the ASCB. I just completely forgot about it.
Silly sausage.
Phil, what are you working on in your postdoc? Are you also working on apoptosis, on extrusion?
No.
Something different?
So I work on organoid. Yeah, like
Oh, really?
yeah, the development of the skin on organoid. So I will, yeah, it will like change of subject.
Okay.
Yeah.
Is it mechanicsy, like you?
Yep. Still, I'm
courses
still
and
working
stuff?
on mechanics, but on organoid system.
Okay.
See how.
Yeah, organizers are super popular nowadays, aren't they? That's cool. You're brave to go in the new year. So I know a lot of people were postponing moving to America because of the election stuff, right?
Yeah. But finally, it turns out okay.
Yeah.
I hope that after 1 month, more than 1 month from now, then there will be some changes.
Yeah.
Like at least in the social distancing and the restrictions regarding the coronavirus.
Yeah. I don't know how do you know how bad it is in Boston?
Sorry?
Do you know if it's bad in Boston?
In Boston during the summer and winter, fall is still okay, but I think in the recent weeks it's become very bad. I don't know why. Although they follow all the restriction, like they have the contact tracing, social distancing, everyone wears masks. Yeah, I don't know.
Yeah.
I think it's partly due to the Thanksgiving, maybe.
Oh, because everyone was moving around just recently.
Yeah.
Yeah. Probably Christmas as well, though, I guess.
Yeah, this Christmas gonna be quite sad.
Yeah. Okay, I've finished all my admin stuff. Okay. Normally there's like 5 or 10 people that will sign up like right now. I have to send them emails.
Oh yeah, I just saw your reviews.
Oh yeah.
Yeah, congratulations. I haven't read it. Yeah, but I will. Yeah.
It's supposed to be part of mechanobiology special issue.
Yeah.
But it's a bit weird because I think the journal was kind of like releasing them. I guess it's not publishing next month, but they're releasing them. It's supposed to be as part of the ASCB week.
Oh, okay.
Yeah.
Oh, because of the AS.
Yeah, they were supposed to like make it joint to that. I think they're being released as and when.
Hello.
Hey, how are you?
And you?
Yeah, good, thanks.
A bit nervous, but you know.
This is the first Zoom summit talk you're giving.
It's the first time that I'm going to present my work, so.
Oh, okay.
Yeah, it's a kind of exam.
Yeah. I think it's a bit weird on Zoom because you won't. I can ask people, should I ask people to put their cameras on so you have faces or you don't mind?
No, I prefer don't see anyone.
Okay. I'll ask people to turn the cameras off then.
Yeah, much better.
Can you remind me how to pronounce your name properly? Because otherwise I'm going to say it wrong.
Besai.
Besai. Okay.
Besai.
Am I saying it right? Besai.
Besai. Yeah, right.
Okay. I'm going to get it wrong.
Don't worry. You are the one that don't know how to say correctly, but all my life was the same.
Oh, really?
Yeah, because the people say Besai, Besai. So yeah, but it's okay. Don't worry.
Okay. I'll say it just once then. So otherwise, I'll just say he. They. You want to? Yeah, check your presentation. Yeah, go for it. How's everything at King's?
Good. Just slowly because the pandemic.
Yeah.
But
are you able to be in every day now? Or are you still on?
Yes, but we have a limit of people working at the same time. So we have to organize and book where are you going to be.
Yeah.
Yeah. So
just stay in the microscope room.
Yes. So you can see this, true?
Yeah, that's good.
Cool. And then I can, yeah. Oh. Yeah. Cool.
Yeah, all good. Do you have a preference about whether going first or second, or you don't mind?
I don't mind, whatever she prefers.
Okay, then maybe I can go first then.
Okay. Sounds good. Oh, and I think we forgot to ask, I don't know whether Jen asked you, Feng, but do you want your stuff being on the YouTube afterwards? Or do you want us to get rid of it? Because obviously it might be early stuff.
I think it's okay. I think it's okay. It just got accepted yesterday, so I think it's.
Oh, really? Congratulations!
Yeah, finally.
That's good timing.
I don't know. I will ask Brian, but
okay.
Yeah,
we can. I think Jen has a way of cropping the video, so we can get rid of. I know we did it for someone in I think Eric's lab a couple of weeks ago. We cropped off their bit because it was a little bit preliminary.
I know how to do that, so I can do that as well.
Okay, great. Yeah, so just let us know, like, whenever.
I will ask him.
Yeah, no problem.
He's here already in the... yeah, he's here.
Okay.
Brian. No. Or she's fine for me. So, yeah, you can leave in YouTube.
Okay, cool. We can always change it in the future, so if you change your mind, whatever, just give us a shout. Cool. So we'll get started in 5 minutes then.
Sure.
Brian says, how could you talk?
Yeah, that's a good point. Thank you, Brian.
Confidence boost before the talk.
Yes. Oh my God.
Are you post submitting or?
This work? No, not
yet. Okay.
I think we need half year to finish something more close. Okay. For now, look good, but we need, and the pandemic is not helping. So
yeah, absolutely.
Yeah.
Well, half years could be close, right?
I hope for the next year at some point we can submit something.
Yeah.
But yeah, we will see.
Okay.
Yeah.
Yeah, it depends how much access you can get to the lab, right?
Yeah. For now, I have time, so I can do.
Yeah.
I can do with my time.
From where did your work get accepted?
NatureCom.
Yeah. Cool.
Nice, congratulations.
It takes me 1 year.
Oh, really? Just, you mean since submitting to NatureComs?
Yeah, since I first submitted.
It's a long one. And now when does it come out? I guess in still another couple of months.
Yes, it's like, it's a big relief.
Yeah, fair enough.
Sorry, has someone got like a whistling noise happening?
I did hear it, but it went just as you said.
Yeah.
Okay, we'll give it a couple more minutes. Okay, I think we can get started then.
Yeah.
Cool. So, hi everyone, welcome to the last seminar of 2020. We've got two great postdocs to give talks today. As usual, if you want to ask a question, just pop it in the chat and I can read it out to the speakers after their talk, either in the chat on Zoom or the chat on YouTube. Okay, so our first speaker is Anne Fung Lee. Fung did her PhD at the Mechanobiology Institute at National University of Singapore with Ben Waladu and Lim Tree Tek as her supervisors studying collective cell migration and specifically on epithelial cell extrusion. And she's now joining Carl Kohler's lab at Boston Children's Hospital for her postdoc in the new year. And today she's going to be talking about her PhD work on cell extrusion. So take it away.
Okay, sure. Okay, thanks, Adam. And first of all, I'd like to thank Adam and Jennifer and Becky for organizing this series of talk. And there are many great speakers and great talks. So I hope that, you could enjoy my talks today. So as Adam just mentioned, my interest is on how mechanical forces regulate epithelial collective cell migration. And the particular process I'm talking about today is epithelial cell extrusion, which is a process to maintain epithelial homeostasis. And most of the results I'm gonna talk about today is already published on bioRxiv, and I hope that the final text will be out soon. Yeah. So when we talk about tissues, it has one more level of complexity compared to the single cell migration. And when we talk about epithelial, we could appreciate that the maintenance of strong contact with each other and with the substrate is central to maintain epithelial integrity. And even though the composition of the cell-cell adhesion and cell substrate adhesion can vary between cell types, the general consensus is that the adhesion complexes are highly mechanosensitive and interconnected to cytoskeleton. And through these connections, the cell can sense and transmit forces. So when the sheath of cell undergo collective cell migration, the cells within the tissues can sense forces transmitted through these complexes, and through that they can change the shapes as well as the motility. And it is also important to note that during the collective cell migration, the disassembly and reassembly of these cell adhesion and cell substrate adhesion complexes are important to enable the cell movement. So as I mentioned, when the sheet of cells start moving, they can extrude their membrane towards the front. And this formation of the front migration is termed lamellipodium. And another types of mechanism that enables the sheet of cells to migrate is purse string, which is the connection of the actin cables that connect between cells during collective migration, and these cables can be contractile. And this purse string is paramount during during epithelial processes like gap closure and wound healing. And also it is important to note that collective cell migration is also important even with the case of within the monolayer when there is no clear front. And this is particularly related to the process called cell extrusion. So cell extrusion is the process when a dying cell or an unwanted cell is expelled from the monolayer. And through this process, when the cell is leaving the monolayer, the neighboring cells surrounding the extruding cell will start to migrate towards the extrusion site and help to remove these cells. So when a dying cells lose contact with each other and leave the monolayer, it could potentially leave a wound inside the epithelial. Therefore, to prevent a breach in the epithelial, the upwards movement of the extruded cells needs to be simultaneous with the sealing actions of the neighboring cells. And how does the cells or the epithelial do that? So there is one model to explain this is that during the cell's extrusion, and in this case, it is the apoptotic extrusion where the extruding cell undergo apoptosis process. The neighboring cell will try to organize a structure similar to the purse string that surrounds the dying cell, and this structure seems to be able to contract and squeeze the dying cell out of the monolayer. However, in several years ago, the results in our lab shows that sometimes the purse string is not compulsory to extrude a dying cell. So in this example, which is part of the result published in this paper on current biology, which this cells is undergoing extrusion, this is the non-fluorescent cells that's surrounded by the fluorescent cell labeled with actin. And then as you can see, when the cell turns on the caspase and start the apoptotic program, the neighboring cell will start to organize lamellipodium protrusion. And this lamellipodium protrusion close the basal side of the extrusion site. And we hardly see any full purse string formation until the very end of the extrusion, where we can see some actin cables. So it indicates that the different structures other than purse string or purse string ring could actually contribute to extrusion. So therefore, we thought that extrusion seems to be more heterogeneous than we thought, even though the event happens in a very small length scale. Therefore, we would envision that the purse string, which acts in a single plane, may not be sufficient to drive extrusion, especially during the cases when we have the epithelium with highly polarized epithelium. So our question is that whether extrusion can be explained in terms of the mechanical forces, and these mechanical forces can be manifested by different structures that can generate different types of signature forces, like lamellipodia, which can produce protrusive forces, and purse string, which can exert a contractility. And since both lamellipodia and purse string engage in both cell substrate adhesion and cell-cell adhesion, whether this crosstalk between cell substrate adhesion and cell-cell adhesion could regulate extrusion. Therefore, we sought to determine when the relative contribution of each mechanism in relation with cell-cell and cell substrate adhesion, and then we aim to provide a mechanical framework that could explain how heterogeneous extrusion is. So in this study, we use MDCK cells as an in vitro model for the epithelial, and also to distinguish between different actin structures during extrusion, we mix the non-fluorescent cells with the fluorescent cell labeled with actin. So therefore, we can distinguish between lamellipodium and the purse string, and also the purse string between the dying cell and the neighboring cells. And also to control better the conditions and everything, since here onwards, I will only talk about an experiment that use laser induction to induce apoptosis program in the extruding cells. Yeah. So the example here shows time-lapse sequences of extrusion, and we could observe that during extrusion, the neighboring cells first, as similar to the example that I showed you before, extend the lamellipodium towards the basal plane to close the basal part, and then this followed by the formation of actin cables at the apical site. And then it is notable that the actin cables formed at the apical site doesn't form a full multicellular purse string until the very end of extrusion, and rather this actin cable seems to be able to contract by itself during the time course of extrusion. And therefore, we believe that this heterogeneous actin organization really exists and dominates in most of the cases of extrusion. We also try to inhibit either lamellipodia or purse string or actin cable contractility by different drugs. And what we observe is that we reduce the rate or the probability of extrusion within the time frame of 3 hours, which is the time frame for normal extrusion. And then it is also interesting to note that if we inhibit the post-string contractility with blebistatin, some cases of extrusion can still occur, and then the basal site can still be closed successfully, but although the dying cell cannot be removed from the monolayer. So now we want to show that the lamellipodium protrusion is the prerequisite for purse-string purse-string formation later. And then we will also try to show that lamellipodium protrusion is as important as purse-string and is very important for the early phase of extrusion. Therefore, we will try to tweak the cell substrate adhesion. And then we try to eliminate or reduce lamellipodium activity without biochemically perturb the purse-string. So therefore, I decide to use the micropatterning approach. So what we use here is that we make use of this substrate with non-adhesive island, and then we use micropatterning to change the size to print on the substrate with the island of different size. And then we can passivate this non-adhesive island and culture the cell on top. And by this way, we can control by this way, compared with laser-induced cell that we can control the extrusion on top of this non-adhesive island. So as I say, by varying the size of this non-adhesive island, we can segregate purse-string from lamellipodium. So let's say if the non-adhesive island size is larger than the cell size, then extrusion fully depends on purse-string formation. And if the non-adhesive island size is smaller than the cell size, then we can allow the lamellipodium to form first before extrusion, and then the purse-string can be continue the work later on. So we could see in this example. So on the left-hand side is the actin, and on the right-hand side is the label for the cell-cell adhesion. And then we can see that this cell undergo extrusion, and the purse-string is able to form around the extruding cells. And even though the purse-string is fully functional, which is shown by the recruitment of the actin around the extruding site, as well as the enhancement of cell-cell adhesion, the extruding cells cannot be removed from the monolayer, as you see that the dead body of the dying cell is still left inside the monolayer. And then by combining this technique with the traction force microscopy, we can also measure the tension generated by the purse-string during extrusion by looking at the average traction force at the edges of this non-adhesive island. And I will come back to this point later when I talk about the generic model. So indeed, when we reduce the size of the non-adhesive patch, we can see that there's an inverse relationship between the size of the patch and the probability of successful extrusion, which further strengthen our point that early protrusion by lamellipodium is crucial for producing enough forces for extrusion to happen. And then we hypothesize that this can reduce the workload applied by the purse-string. And then one more interesting observation we have is that when we look carefully at the purse-string organization around the extruding site on top of this non-adhesive island, we observe that there are some radial cables that emanate from the purse-string structure, as well from the cell-cell junction that connect between the cell-cell junction and the cell substrate adhesion. And then this radial cable seems to be persist during extrusion, which we guess that this can contribute to the resistivity of the neighboring cells during the closure of the extruding site. So we shows that the heterogeneous actin structures is important, and the lamellipodium protrusion contribute to a lot of workload for extrusion. And next, we want to question whether the presence of the purse-string is dispensable. And one way to approach this is that we will interfere with the cell-cell junction, as cell-cell junction is important to maintain the purse-string. So we look at the extrusion of the cells from the monolayer of alpha-catenin knockdown cells. So when we knock down the alpha-catenin, we can reduce the cell-cell junction strength, and by this way, the purse-string can be compromised. And indeed, when we look at the extrusion of cells from the monolayer of alpha-catenin knockdown cells, you could see that the extrusion is fully engaged in lamellipodium formation. And even though there are some actin cables that form during extrusion at the apical site, when we perform laser ablation on these cables, we see lower contractility as compared to the wild type. However, one surprising thing we note is that even though the extrusion here depends fully on the lamellipodium formation and lack of pulse string, contractility extrusion can still occur within the time frame of 2 hours. And if we rescue the alpha-catenin knockdown with different alpha-catenin mutant with enhanced reduced cell-cell junction strength, we could change the profile of mechanism related to either pulse string or lamellipodium. But in overall, it doesn't change the time scale of extrusion. Therefore, I was quite intrigued about it, and then I questioned, well, other than pulse string and lamellipodia, which contribute to the forces involved in extrusion, what are the other parameters that can interfere in the model? And therefore, therefore I collaborate with Jean-François Roubaix, who is now in Century here, for outlining a theoretical framework to put in the parameters important for the forces that regulate extrusion. So we simplify the extrusion cells as a small gun of diameter R0, and then we can write down the forces at the margin of the extrusion cells by this equation. And all of these parameters are the one that that are based on our observations, and we are also able to verify each of these parameters in the experiment. And then you can find the further analysis, the detailed analysis in the full text in the bioarchive. But I will go through the general parameters here. So the forces or the stresses at the margin of the extrusion site depends on several parameters. And the first one should be the forces exerted by the lamellipodium at the margin of the extruding area. And the second one is, of course, the pulse string, which generates a kind of light tension. And also in this equation, we put in the viscous stress. And this viscous stress, it comes from the bulk resistivity as well as the formation of the apical actin cables that we observed previously. And finally, in our model, we introduced the noise fluctuation, which is a stochastic parameters. And if we solve this equation, for R as a function of time, we can show that the extrusion probability extrusion can still happen in the time scale of 150 minutes, given that the forces exerted at the margin of the extruding cell, as in the lamellipodia component here, is positive in a fully adhesive area. And then when we turn to the case where the substrate adhesion is compromised, the equations become a bit different. So in this case, the sigma p, which is the protrusive forces, become negative. So therefore, it contributes to the resistivity of the bulk. And then also we for this parameter, which is the viscous stress by the bulk, it adds in the component that there is the resistivity of these radial cables that I introduced previously, that contribute to this viscous stress that drag back the wound areas. And then also as I mentioned, we use we combine this micropatterning with traction force microscopy. So therefore, we can measure the tension, the effective tension that's generated by the pulse string. And then when we compare between the wild type and the alpha-catenin knockdown, and indeed we can give the estimated values for the tension. And then these values can be used later on for our numerical simulations to compare between the model and the experiment. And finally, we use the as I mentioned, we use the numerical simulation that use the parameters that we can measure in the experiment to solve the probability of extrusion as a function of initial size of extrusion cells, as well as the pulse string contractility. And then we can compare our simulation with our experimental results, where we can make use of different alpha-catenin mutant cells on top of all of these non-adhesive island. And if we come, and then we can see that the trend obtained from numerical simulation and experimental results closely follow each other. And then we can also play with the fluctuation parameters. Then if we increase the fluctuations, we can actually shift the phase diagram of probability of successful extrusion towards more higher percentage. Therefore, our analytical model demonstrated that the presence of fluctuation may lead to the completion of extrusion process, and even in the space of parameters where the contractility or pulse string force is limited. So therefore, it can explain partly why in the fully adhesive substrate, when we use different kinds of alpha-catenin mutants, the extrusion can still happen within the time frame of interest. So in short, we show that extrusion really depends on the combination of forces from lamellipodium, protrusion, and pulse string contractility, and all of these could be fine-tuned by cell substrate adhesion patterns and cell-cell junctional strength. And also we provide a holistic framework that explains all of this heterogeneity of extrusion. And by considering all of these parameters, we could predict when and whether extrusion will fail or succeed. Yeah, so so that's the end of my presentation, and I would like to thank my supervisors Benoit and Citi for helping me during my PhD, as well as my collaborator Jean-François, who is now in Century. And I would also like to thank Yusuki and Renemark and as well Lo Bun Chuan for helping me during my PhD, as well as the transition period. And yeah, and I would like to thank all of my collaborators and co-authors. Yep.
Thanks, Fang. That was really nice work. So any questions, please pop them in the chat. I'll just start off by, I was wondering, and I might have missed this, are you able to compensate for low cell substrate adhesion force by increasing the force of the pulse string?
We could to some extent, as in here. So if we use the alpha-catenin delta mod mutant, which has constitutively active vinculin, which can enhance the cell-cell junctional strength, it can compensate a bit. Yeah.
Okay, but you need some cell substrate force, I guess, to. It's not completely redundant.
Yeah, it's not completely compensate.
Cool. So we have a question from Oscar Ruiz, who says, I'm curious about cell autonomy of each component in extrusion. Have you tried making mutant knockdown clones? So instead of having all the cells knocked down for alpha-catenin, maybe only the extruding cell is mutant.
Yeah, I did, and then it still occurs within the correct time frame. Yeah, so I did try different mix of alpha-catenin knockdown and Y type, and even like with the neighbors, some of neighbors are Y type and some neighbors are alpha-catenin knockdown, and they still do their job. So alpha-catenin knockdown, they still extend lamellipodium, and the Y type, they still form the actin cables, and extrusion can still be complete. Yeah.
Great. Question from Andrea Ravasio. Says, hi, Feng, nice work. Does the confluency of the cells influence the heterogeneity or stochasticity of the process?
Yes. So yes. So in most of the experiment, I try to keep the confluency at low, when it start to cross from the low density to high density, and in the model there is the parameters that depends on the height of the monolayer, and the height of monolayer is partly due to the confluency. So yes.
Great. Parissa asks, what role do basement membrane and focal adhesion play in your model?
That's a good question. Actually, in our model, we just we consider in terms of the forces, but for focal adhesion, we assume that the focal adhesion turnover is at smaller time scale than um, the formation of lamellipodia of a purse string. Yeah, so so in our model we didn't really consider this.
Thanks. Christina Bertocchi says, so first the lamellipodium is formed, formation, and then the purse string. Shouldn't we expect to see focal adhesion at the front during closure?
Do we expect the focal adhesion to form in the front? Yes. Yes.
Yeah.
Yes.
Okay. Maybe if Christina wants to follow up. So one more question from me. So does the signal that causes the apoptosis matter in terms of the model? Or is it just the fact that you know that the cell is being extruded and it doesn't matter what's caused it?
I think it's yeah, that's a good question. So if we talk about the neighboring cells mechanism, then I think that the neighboring cell just says that there is a change in the tension between themselves and the extruding cell. So it so it affects the mechanism. But if we talk about the extruding cell itself, then maybe it has a bit different. And then I believe that some other ways of extrusion. I mean some other ways of apoptotic, and it may not be able to form the purse string within the dying cell itself. But that is not in our scope of this research.
Cool, thanks. Christian Camarota says, when MDCK cells are overpacked, they induce extrusion to return to homeostasis. Would this increase the viscous resistance in your model, and would that affect the probability of successful extrusion?
Could you repeat the question?
So when MDCK, sorry, I forgot, I said packed, I meant overpacked. So when MDCK cells are overpacked, they induce extrusion to return to homeostasis. Would this increase the viscous resistance in your model, and how would that affect the probability of successful extrusion?
So when it is overpacked, yeah, that's a good question. I think that the viscous stress components, the sigma mu, as I mentioned previously, it depends highly on the height of the monolayer, and then I think the overpacked MDCK could affect. Yeah, it could increase the viscous stress. Yeah.
Okay, great. I think we've covered all the questions. So thanks very much, Fen. We'll move on to our second talk today.
Okay, thank you.
So second speaker today is Besai Sanchez. Besai completed his PhD in Seville studying macrophage migration in Drosophila embryo at the Andalusian Center for Developmental Biology in the Martin Bermudo lab. He moved to London in 2016 as a postdoc in the Stramer lab. He is studying macrophage behavior during Drosophila embryogenesis to analyze how the cytoskeleton is regulated through cell migration and how cells build the extracellular matrix during development.
Hi everyone, I'm Besai Sanchez, postdoc in the Stramer lab, and I would like to thank you, the organizer, for this opportunity to share my project about the interregulation between the cortical and lamellar actin network. Let me let the laser. Cool. So here is an example of a fish keratocyte where we can see a big flat region of the cell called lamella. This region, we can observe an actin network, and as you can see in this diagram, this network is cross-linked and organized by a number of components such as myosin, ARP23, or cofilin. The actin in the lamella is dynamic and present a retrograde flow from the leading edge to the rear. Here you can see an example of an in vivo. So as many of you know, the actin flow has been extensively studied because of his role in regulating cell migration. However, there are still many questions that remain unanswered. So to address some of them, we use the embryonic Drosophila macrophage as a model system. In this movie, you can see a ventral view of the embryo and macrophages dispersing during development. Macrophages are born in the head and migrate through the embryo. At this stage of development, we can study the macrophage migration by observing their movement close to the embryo surface. Here, we observe the cell dispersing evenly and organizing in 3 parallel lines that you can see in this example. 1 line, 2nd line, and the 3rd line. So we can obtain high-resolution imaging in vivo, which allow us to study the actin flow during this cell-free movement. Here is one example of a high-resolution movie that we can obtain. You can appreciate his migratory behavior and how the actin flow is evident in the lamella. These cells have a large lamella that our lab has been extensively studying, and a cell body where the nucleus and the organoids of the cell are located. By characterizing the actin flow in this kind of movies, we recently published this paper, in which we show how a persistent and polarized global actin flow is essential for cell migration. To quantify the actin flow, we use the particle image velocimetry, PIV, which allow us to track the actin movement in the lamella. I don't have enough time to show more relevant data from this work, but I would like to highlight 2 important results. So the first one is that we found that myosin has a gradient across the lamella, producing a polarized distribution of this protein. Myosin localization in this way has been described in other cell types. Also, using the data from PIV analysis, we can study the compression in the actin network. As you can see in this graph, there is a peak of compression at the rear of the lamella, so in this part of the cell, suggesting that this part is important in the flow regulation. In order to visualize that compression, we developed this tool, where we can see the accumulation of deformation in the actin network. This analysis highlights great compression in an area around the cell body. So as you can appreciate in this zoom, if we look at the actin localization, we can see a high level of actin in this area. So we're starting to think that maybe there is an actin cortex. The actin cortex is another actin organization, different to the lamella, classically described in cells undergoing meiosis or mitosis. In this diagram, you can see an example of the actin cortex organization, where actin is highly connected with the cell membrane through some specific cortical proteins, as the protein in the erythropoietin myosin family. So we decided to check how this component defining the actin cortex look in our system. So we took high-resolution image with different GFP reporters, so live actin to visualize actin, myosin wild type to visualize myosin, and the PIP2 binding site of the PLC protein to visualize PIP2. And as you can see in the image, the region around the cell body of this macrophage is decorated with all the 3 components. So here you can appreciate the actin, here is the myosin, and here is the PIP2, suggesting that the presence of an actin cortex. So therefore, we decide to further investigate myosin. An important property of Drosophila as a model system is that the genetic is relatively simple. So for example, myosin is the only protein in Drosophila of the ERM family. So this protein is described as cell cortex organizer, and it is highly conserved between organisms. In particular, the site of phosphorylation T559 is conserved, and this controls the myosin activation. Myosin is also regulated spatiotemporally by PIP2, a phospholipid present in the cell membrane. It is unclear if myosin needs to first recognize PIP2 in order for the phosphorylation to happen, or if these two processes are independent. So we can explore the genetic tool available in Drosophila to decide Myosin activation, and we are going to explore them in this project. So using the Galford system, we can express different versions of Myosin, specifically in the macrophages. This diagram shows the full length of Myosin, where we can see the PIP2 binding site and the T559 phosphorylation site. Both of them can be mutated. So we start with the Myosin wild type, which work as the endogenous Myosin present in the macrophages. We can study the role of PIP2 using one version of Myosin without PIP2 binding site. Moreover, we can also study the relevance of the phosphorylation site, expressing one non-phosphorylatable Myosin, or the opposite, a phosphomimetic Myosin. Finally, we can combine this version of Myosin with either PIP2 site and the phosphorylation site mutation, having a non-phosphorylatable Myosin without PIP2 binding site, and one phosphomimetic version of Myosin without the PIP2 binding site. So just to recap, we have one Myosin wild type, which represents the full endogenous Myosin in macrophages. We can affect the PIP2 binding site, having a full version of Myosin without PIP2 binding site. Moreover, we can mutate the phosphorylation site, obtaining always inactive Myosin or always active version. And also, we can combine the phosphorylation with the PIP2, having another two lines combining these two mutations. So here we're going to start comparing Myosin localization in various versions that I just introduced. So here you can see again how look Myosin wild type. In this case is how look when the PIP2 binding site is removed. In this case, is followed by the non-phosphorylatable Myosin version, and finally the combination of both mutations. And you can see the distribution look different at the cortex, as you can appreciate. For example, in this case, there are less intensity, but we are still working in the quantification. So today I will focus at the lamella. So we quantify the intensity across the lamella, drawing line scan from the cell edge to the cell body. So as you can see in this graph, the quantification confirms that similar localization between all these versions of Myosin. Now, we're going to see the opposite. So we still have the Myosin wild type without PIP2 binding site, which we are comparing with the phosphomimic version of Myosin, so it's always active. And I hope you can appreciate how different localization of the phosphomimetic version look compared with the control. In fact, we observe a gradient with more protein located at the rear of the lamella, as you can see in this graph. Interestingly, when we remove the PIP2 binding site from this phosphomimetic version, the protein distribution across the lamella changes too. And as you can see in the graph, a gradient is still present, but less pronounced. This suggests that the PIP2 has a role in the phosphorylated Myosin localization. And this gradient across the lamella reminds us the Myosin gradient that I showed you previously. So we started questioning how this gradient is forming. Some papers suggest that the actin flow can regulate this protein gradient. And I'm showing here an example. So the idea is that the localization of protein with actin affinity can be regulated by the actin flow, thanks to that tension across the cell. This spontaneously set up a gradient across the lamella, as we and others observe for Myosin, and as just showed you for the phosphorylated version of Myosin. So we hypothesized that the ability of the phosphorylated Myosin to have a gradient is related to an enhanced actin affinity. So in order to investigate this, we decided to make some FRAP experiment to study actin affinity of the Myosin lines. And you can see in this short movie, and as previously described by other group, when we bleach in live actin, we observe almost instant signal recovery, despite the high frame rate of 0.5 seconds used in this case, as the proof is the constantly binding and unbinding. Something similar is observed for Myosin control, with the fluorescent signal coming back quickly. However, this is not the case with the phosphomimetic version of Myosin, which display a slower recovery. So in the following panel, we summarize the FRAP experiment. So you will have a pre-bleach picture, a post-bleach, and then 7 seconds after and 40 seconds after. In the top row is the Myosin control, and the recovery is very quickly. And therefore, you can really appreciate a black region in the ROI. And it's confirmed by the quantification, as you can see in this graph, the recovery intensity. Now we'll look at the version of Myosin without PIP2 binding site, and we see something similar, with a quick recovery, as you can see in this cyan line. The next row is the non-phosphorylatable Myosin, so always inactive, and doesn't show different. We can then, and the last one is the combination of the non-phosphorylatable and the without the PIP2 binding site, and that doesn't have different neither. So we can calculate then the protein half-life using the signal recovery, and as expected, they are not different in the protein half-life, as you can see here. Now I would like to show you the phosphomimetic version of Myosin. We keep the control and the PIP2 binding site for reference, and here, as I mentioned before, you can easily see the bleach area over time, as the recovery is slower, and you can see in this blue line. However, in the case of the phosphomimetic version without PIP2 binding site, the speed of the recovery is rescued, similarly to the control, and you can see in this teal line. By quantifying the half-life, we found that only the phosphomimetic version of Myosin has a slower diffusion. So this experiment suggests that the phosphorylation increased Myosin affinity for actin, but also that PIP2 is also involved in regulating Myosin diffusion. It could either enhance the strength of the actin binding, or it could modulate the interaction with the cell membrane. So all these experiments support the idea that the actin flow is controlling the Myosin localization. And Myosin is likely candidate to be regulated by the actin flow thanks to its high actin affinity. Now, I would like to show you some preliminary FRAP results, but this time the ROI is located at the cortex, as you can see in this example. And here is the graph, and I hope you can see that there is no difference in the Myosin wild type if we compare lamella with the cortex. On the other hand, surprisingly, the phosphomimetic version of Myosin has an even lower diffusion at the cortex, as you can see, appreciate in this graph. This suggests that the phosphorylated Myosin has more affinity to the cortex, likely because of the localization of PIP2, which is enhanced in this region, as I showed you earlier. Seeing this result, we decided to also investigate the role of Myosin during macrophage migration. As you can see in the bottom movie, macrophage dispersal is affected in the Myosin mutant. There are large gaps between macrophages, and the 3 canonical line distribution is lost. Moreover, the macrophage speed is also affected with a slower speed compared to the control, as you can see in this quantification. When we look at freely moving single cell, and we apply our tools to quantify the actin flow, we observe something very interesting. So the absence of Myosin affects the actin dynamic, reducing the flow speed. This is surprising, and we don't understand this fully yet, but it suggests some sort of feedback between the actin and Myosin. Not only the actin flow regulates Myosin localization, but also Myosin at the rear of the lamella can regulate the actin flow. So I would like to summarize the key findings of the project in this slide. So the macrophages have a cortex decorated by actin, Myosin, and PIP2. The phosphorylation state and PIP2 regulate Myosin localization and dynamic. And also, the absence of Myosin affects the macrophage migration and the lamellar actin flow. To put this in context with other work, it has been previously shown that proteins in the ERM family, such as Myosin, are important for cell migration. Moreover, it is it was observed that such protein localized at the back of different cell types during migration, as you can appreciate in this picture. However, it is unknown how these proteins are going to the cell rear, and it is not clear how mechanistically they control cell migration by localizing there. We hypothesized that the localization of myosin at the back might be related to the actin flow. So this work that I mentioned earlier shows how the actin flow can regulate the localization of any protein that binds actin, generating a polarization across the lamella, thanks to the advection. They focus on the removal of negative polarity factor from the cell, from the leading edge. However, a similar behavior could be related to the localization of protein at the rear. Our hypothesis is that the actin flow is a critical regulator of migration. The actin flow can integrate a number of behaviors and spatiotemporally control the distribution of polarity factor across the cell. Therefore, actin flow can regulate all kinds of polarity factors, not just the one at the front. Actin flow can regulate myosin as it binding actin, and the possible feedback between flow and polarity that they hypothesize may be relevant also for factors at the back, like myosin, which could in turn control actin. Interestingly, we show how macrophages present both a lamellar actin flow and an actin cortex, which can be considered unusual as cells normally are thought to display only one of the two actin organizations. But recently, this observation has been challenged by this work where a new tool was designed to locate the membrane proximal F-actin. This tool highlights the cell membrane associated with actin and therefore the most superficial actin cortex. Interestingly, the author showed many cell types that were not thought to have an actin cortex displaying signal for this new proof. Moreover, they highlight how actin associated with cell membrane co-localized with a phosphomimetic version of enrin, another ERM protein. This suggests that there could be more cells with both actin organizations, and that macrophages might represent a valuable model to study the interregulation between lamellar and actin network. I would like to say thank you all you for your attention, and thank you Brian for his support, and also Stefania and Eduardo for their help with the image analysis, and NC for her amazing diagram, and of course the rest of the Strammer group, and again all you for your attention. Thank you so much.
Thanks, Besse. That was a really nice talk. So we'll kick off with. Sorry. Yeah, you're quite a little fast, but don't worry about it. So a couple of questions from Oscar Ruiz. So the first one, for your macrophage migration assays, do you use the GAL4 in a Myosin mutant background, or is there wild type Myosin around?
No, it's in the wild type background. We are still working to see how look all these different Myosin lines in a Myosin mutant background, but it's tricky because we have to play with the Drosophila chromosome, but yeah, we are working on that.
Cool. And a second question from Oscar. It looked like there might be less macrophages and they looked bigger in the mutant Myosin. Have you quantified this?
Yeah, we quantified the cell shape and we didn't see different. Now we are going to check just cell body or lamella to see if they are different or not. But for now, in average, they are not different.
Thanks.
Hey, sorry, I missed the beginning of your talk. I had a meeting that I couldn't miss, but I'm glad that I was able to catch the end of it. So we have a question from Juanma. Thanks for the talk. Is the cell migration persistence or polarity perturbed in the different Myosin mutants?
In the Myosin mutant, we didn't see a problem in the actin organization at the lamella. And now we are overexpressing all the different Myosin lines, and we are going to check if that overexpression affects the actin organization. But the Myosin mutant doesn't produce any phenotype. in the actin organization, just in the speed.
And since I missed the beginning, maybe you can just help give me some insight. Do you have a sense of this being a really generalized mechanism, or do you think it's more a little bit specific to sort of the types of cells that you're studying?
No, we think it's something that we can check in other cell types. The only good thing is the Drosophila, there are less genes, so it's more easy to check the mechanical. But we are planning to check misshapen, sleek, some protein that phosphorylate myosin, to have the full history with the all regulator of myosin. And yeah, but yeah, I think it's something that is conserved because this family is very, very, very conserved through the development. So this myosin, the phosphorylation site is highly conserved. It's almost the same in all the organisms. So yeah.
And do you see changes in the migration speed when you change the myosin mutants? Because that's changing the actin flow. Do you see a change in the speed or directionality?
In the myosin mutant background, we saw a slow speed in the macrophages. So yeah, and we didn't see a high phenotype when we overexpress the different myosin lines. Just we are working in that, but for now we didn't see any relevant phenotype when we overexpress, because the macrophage has the endogenous myosin, because everything is in a wild type background. So maybe when we use the myosin background and then we overexpress all the myosin lines, then maybe we can see rescue phenotype, no rescue, or other kind of phenotype. Yeah.
Great. We have another one from YouTube. Nath House asks a more general question. Are these mutants able to develop properly?
No, they died in the embryo. The Drosophila have embryo stage, then larvae, and then adult, well, pup, and then adult, and they died in them at the end of the embryogenesis.
Thank you.
Sayali asks, do you see differences in actin polymerization and recovery rates in the phosphomimetic mutants?
We are doing, yeah, it's a good question. We didn't know yet, but we're working on that. Yeah. The thing is, with the pandemic, we had to organize the work, and then we are more slowly doing the different experiments. So yeah, but yeah, of course we are going to check that.
Yeah, it's amazing how much everything has really slowed down a lot.
Yes.
Amazing. Yeah, awesome. Thank you so much. If anyone else has any questions they want to pop in, this is your last chance. Feel free to.
No, they can send me an email if someone thinks something later and say, oh, or contact us by Twitter or the website or whatever. Yeah.
Perfect. That's great. That's good to have on the record that you're available. So, okay, I think that's actually it. So we're officially wrapping up for 2020. It's been a crazy year, but I'm really, really glad that we were able to spend it with all of you wonderful people. So thank you so much to our two speakers today. Again, apologies from me that I had to miss most of this, but I was glad I was able to jump in at the end. So just so you guys know, we are putting together a schedule for 2021, and we intend on continuing to be here to serve all of your cell migration needs. So thanks again to both speakers, and then to all the speakers that we've had for the nearly 30 talks that we've been able to host this year. So we'll see you guys in 2021. Bye.
Bye-bye.
Thank you, guys.
All right, Adam, now you have to end the live stream.
I don't have that ability.
Whoever's the host has it.
Becky.
Becky.
Oh no, I have to make her the host. Okay, so she can. I don't know how to do YouTube.
It's in the lower right-hand corner if it helps.
I'm gonna make Becky the host so she can do it. Happy Christmas, everyone in the chat.
So y'all Brits don't say Merry Christmas. You say Happy Christmas, huh?
You can say Merry. You're taking it away too. Yeah. You notice the smallest thing.
You're my gateway into understanding.
Yeah.
All right.